Okay, let me welcome you to the afternoon session. Our session is on AI in health. I'm Lydia Kavraki and I will be chairing the session again. Let me start the session by saying that health is arguably a topic that concerns all of us. Developed countries spend 10, maybe up to 20% of their gross domestic product on health. In underdeveloped countries, lack of health information and lack of health care is a matter of life and death. It's not surprising that AI is now used to process the vast amount of health data that we are accumulating through the literature, electronic health records, and modern experimental studies, as well as shed help in the design of new therapies. Today, we will listen to talks that show examples of how AI systems have been built to address biomedical semantic indexing and question and answering, advances in medical imaging and precision medicine, and recent efforts in deciphering complex structures such as the visual cortex. The first speaker is George Palouras. George is a research director and head of artificial intelligence lab SCALE in the Institute of Informatics and Telecommunications at the National Center for Scientific Research, Democritus. He holds a PhD from the University of Manchester on machine learning, and he has worked on the area of AI for the last 30 years. He has served as a board member in national and international scientific societies, has an extensive publication record. He has been on editorial boards, chair of international conferences, a very distinguished career. He's also involved in many European and national research projects, let me mention BioAsk, a challenge on large-scale biomedical semantic indexing and question answering, and HIAS, integration and analysis of heterogeneous big data for precision medicine and suggested treatments for different types of patients. So, with this, let me welcome George.
Thank you, Lydia. Thank you, everybody. Thank you for inviting me to this very interesting workshop. And thank you for coming back from lunch. This is nice of you. I'm going to try to give a slightly different talk from the previous ones, or from most of the previous ones, in the sense that I'm not going to be talking about our own research, but I will try to motivate people to spend some of their time in helping other researchers do the work. In particular, I'm going to talk about a project that we have been running for more than 10 years now, on the task of biomedical semantic indexing and question answering. And let me spend a few seconds in explaining why this is an important problem. The main issue that I think is fairly obvious to all of us is that it is important that all this knowledge that is generated by biologists and life science researchers is accessible. There is an enormous amount of information that is generated. Luckily, it is provided centrally now in PubMed, but that doesn't mean that everybody who needs it has access to it. The doctors or the medical researchers that would be able to work with it have a very hard time finding the information that is needed. So it is worth trying to do something about it. And we are doing quite a bit in terms of generating new predictive models, generating language models and so on for this task. But 10 years or 11 years ago, we decided to also try something different. What can we do to mobilize the researchers around the world, the AI researchers, to work on the topic? And for this, we organized a competition, and we call it BioAsk. So BioAsk is now a series of challenges or competitions. It has been happening every year for the last 11 years now, and it has a number of shared tasks. The first task, that we call Task A, has to do with biomedical semantic indexing, so putting labels to documents. The second task has to do with answering questions, and this has become very popular nowadays with ChatGPT and all the like. But it's not exactly the same when we move to medical questions. And this part, this task B we call it, has two phases. The first phase, we call it the information retrieval phase, is bringing back the abstracts and the sentences from those abstracts that are more related to the question. The second part is to actually try and compose the answer. And in addition to those two basic tasks, we have been experimenting with a number of other tasks, and I'm going to be briefly talking about them at the end. But let me also explain how all this started. So luckily we had research funding from the European Commission when we started the project to set up the infrastructure and the ecosystem that you can see in this slide. At the center of this is the data that is generated. And there is data for different tasks, for the different tasks on the left. Now, to build this data, we depend a lot on existing sources, like PubMed and Medline, which is kindly provided by the National Library of Medicine. We also had the chance to build tools for experts. We built a small team of biomedical experts, so doctors, biologists, and so on, that were using the tools that we gave them to generate data, to generate interesting questions. And we have always had an emphasis on real-life, interesting research questions for the experts, not artificially generated questions that are interesting for the AI researchers. So with this, we come to an interesting data set, and this is, if you want, the backstage, the top of the figure is what is happening backstage. And at the bottom, you see the front end, which is the actual, how we run the actual competition. We provided a platform. The participants were able to submit their results and get leaderboards and all the things that you are familiar with. So all this infrastructure was generated in a European project, and then we are maintaining it since then. And throughout the years, we had the honor to have many different teams participating in the competition from around the world, from all over the world, more or less. More emphasis, of course, in developed countries, as one can imagine, but some representation from developing countries, and also from academia and industry. And we think that this is an important result on itself to mobilize all these people. Let me give some basic information about the tasks that we were running. The task A, the semantic indexing task, is heavily based on the abstracts that are coming out every week, every day in Medline. We give the participants some of those abstracts every week, and we ask them to generate labels for them within 21 hours. And the most important point here is the third bullet in the slide, which is that it was getting the right labels, getting the truth, if you want, was very cheap for us because the National Library of Medicine did that with manual curators anyway. So we were very fortunate that we could evaluate how well the systems did without spending any resources on it, other than what an LM spends anyway. And this is organized in batches of 5 weeks, 3 batches, and so on. An example here, you see an abstract. It belongs to the class drug interactions. I should mention here that each abstract actually belongs usually to more than one class, so it gets more than one labels. And this comes from the MESS hierarchy, which is a very big hierarchy of labels. Very interesting hierarchical multi-label classification task. And you see here the numbers that we have. The training data has been increasing since we started in 2013. It's now up to 15 million articles. We get about 30,000 labels. And as I said earlier, on average, we get 12 point something labels per article. And every week we gave about 6,000 articles, which means that about 150,000 new articles every year. These were the test data that we're testing the systems with. And every year the test data is then incorporated in the training data for the next year. Participation over the years, you notice here that it finishes in 2022 and not in 2023. This is an interesting, it's not a mistake, it's an interesting event that I will be explaining in a bit. Participation has been rather stable, around 10 teams on average, not the same teams every year, of course, and several systems. Each team can participate with more than one systems. Regarding the approaches, we have experienced the move from simpler bag of words representations and support vector machines, nearest neighbors and so on, to the deep learning era and the neural embeddings and so on. And of course, this has resulted in increasing performance in the accuracy of the systems. This is a very interesting figure that makes us, it's very rewarding for us because it shows how the system started very low at 0.55 in terms of F measure and how they moved. So the red line is the top performing system each time. So the top performing system reached more than 70% in terms of F measure. More than the red line, the green line is what is very interesting. The green line is what NLM is calling the MTI system. This is the system of the National Library of Medicine that is used by manual curators as an assistant. So this system is actually recommending automatically what the label should be for the document. And as you see, the performance of the system has increased by more than 10 percentage points. And this is due to the collaboration of NLM with the participants of the BioAsk challenge. So the way in which the NLM worked improved through this collaboration. And this has a result to the interesting event that I alluded earlier to. In 2022, the NLM decided that manual curation was not needed anymore, and that they go fully automatic. And to us, who have experienced this change over years, it was sort of expected because we knew that the inter-annotator agreement between humans was actually not much lower than the performance of the systems. So we think this is a high point where we can say that the work that we have done has contributed to an interesting development. So this has to do with the semantic indexing task. We're getting now to the harder task of question answering. So we get now our experts, to set questions, and the systems try to answer these. We release a few questions, about 100 questions every 2 weeks, if I remember correctly, and we request in 24 hours to have the answers to those questions. The documents that are related to the questions. Then we give to the systems the documents that the experts consider relevant 1 day later, and the systems are asked to generate the actual answers. The evaluation here, unfortunately, is not as cheap as in the first one. We have some automated evaluation based on the golden answers that the experts provide, but we also have manual assessment at the end by the experts. And we have different types of questions, from the simpler kind of questions where you have yes or no answers, to questions that have to do with facts, like where is a particular protein localized, or lists of facts, and the more difficult ones where you actually have to generate a narrative answer, very much like ChatGPT is doing, for instance. Over the years, we have built an interesting data set that is now used by many, many people. It approaches now 5,000 questions. that has been created by the BioAsk experts, more or less even distribution across the different types of questions. And we have 4 to 5 batches every year, as I mentioned, that correspond to 350 to 500 questions every year. Participation has been higher for this task than the previous one. We have about 20 teams participating every year on average. Again, not necessarily the same teams, and more systems that participate. And as you can see, of course, this task is continuing in 2023 in contrast to the previous one. Regarding the methods that are used, I think the change in methods is even more impressive in this task. We started by pipeline methods with many different modules and components based on bag of words and ontology-based name entity recognition and so on. And we have moved to end-to-end neural networks more recently. In some cases, combined with ontologies and knowledge graphs to improve performance. Here you see again one of the result figures that we have for this task. And what is interesting here is that the easier type of yes or no questions we now get almost perfect responses by the systems. But also the harder questions where we actually have to get the facts as answers. This is also improving and has improved quite a lot to a very decent 60 or 70% in terms of F-measure. So there is a stable improvement that also satisfies us as organizers to be able to contribute that. So wrapping up this part as well, what I consider most important for this task, with the most important contribution of the BioAsk competition in this task, is actually building the community. So there is actually a community of AI teams around the world that know that every year there will be a new BioAsk competition where they will go and test their systems and they get objective results of how well they're doing, using real questions from real biologists and medical researchers. This is for us very important. At the same time, as I said, we have a unique data set that is being used by academia and industry. It has also been used to generate a version of GPT which is now called BioGPT. And we get this improvement in performance that is also very interesting and rewarding. Now moving away from those two main tasks that we had in the competition, more recently we started introducing another task which we call synergy. So if you think about the task that I have just described, task B, the question answering task, we have a serial process of 3 stages. The experts generate data, questions and golden data. We give them to the systems, and the systems have to produce results. And then the experts come back to assess this. And this is it. It ends. This is fine when you have questions where the experts know the answers to. But what happens if you have questions where the answers are not known? These are the more interesting open questions. Like in COVID, for instance. During COVID, there was we were running like mad to trying to answer questions that nobody knew the answer to. And we had a lot of data, a lot of knowledge in publications, but nobody could put it together to answer those questions. So we said, what if we try to leverage on the community that has been created for BioAsk to try to answer this kind of open questions? And that's why we created Synergy. So Synergy is a more interactive competition, if you want. I don't even know if it should be called a competition, to be honest, where the experts pose questions that they don't know the answer to. The systems try to answer those questions. The experts come back and evaluate the responses of the systems, but they also modify the questions. So they can actually modify their own questions. So there is this interaction between the experts and the systems. And you see this just samples of yes or no questions. that we had. This is still at an experimental stage, this synergy task, but I think that it can have very interesting results and side effects if you want. In addition to that, we had a number of other interesting tasks that we have worked on during the years. We worked on finding funding information in documents that was of interest to NLM, and we have a stable collaboration in the last few years with the Barcelona Supercomputing Center to work with Spanish documents, articles, patents, case reports, and so on, written in Spanish. And we are very interested in expanding that also to other languages. We are in discussions about Portuguese, about Russian, about different languages. So wrapping up and coming to the end of my talk, there is, I hope I have shown to you that through the 10 years that BioAsk has consistently been taking place, it has had a number of interesting results, and it has contributed to research in ways that our own research would have difficulty doing. I'm not saying that we should stop our research, whatever we're doing, but what I'm saying is that spending a bit of time in helping others to do research can actually have a multiplying effect. And it can be very rewarding. But there's also a lot of interesting questions that have come up during the years regarding the tasks that we have been running. In terms of semantic indexing, I talked about already the multilingual and cross-lingual tasks. We're very interested in studying the evolution of biomedical knowledge over time. Do the labels that we give to an article at a particular point in time have to change over the years? We're very interested also in new machine learning methods like learning zero-shot and few-shot learning when we don't have enough data. We're interested in trying to see the connection to knowledge graphs that are becoming more abundant these years. So there are lots of things that we are thinking about doing and try to prioritize what to do next. In terms of question answering, what can I say? We're all experiencing the effect of large language models, and there are interesting questions that we try to answer regarding how applicable they are to biomedical question answering. I'm sure we can get a lot from large language models. I'm also pretty sure we cannot get the final required answer from them. So there's going to be more work needed, especially regarding the trustworthiness of the results. In addition, we want to do some more community-based work. We're thinking of moving BioAsk Synergy, the last task, the interactive task that I described earlier, to a more service-like system. And we're also looking at creating an open source repository, especially for making the results reproducible, but also to be able to build on top of what others have done. This is more or less it. I would like to end my talk by thanking all the different people who have supported us through the years. At the time, we were supported by Google and Ovid being part of Clover, Atipon being part of Wiley, and Plantiel for Spanish. But we started with the support of the European Commission, the National Library of Medicine, and some companies. And especially in the first years, a lot of the work that I talked about was actually collaborative work by many different teams around Europe and with the University of Houston. This is it. If you're interested and you want to learn more about BioAsk, please visit our website. And if you're even more interested, please join us in Thessaloniki in September. Thank you very much.
Take one or two questions.
Okay, great. Thank you very much. Very interesting talk, and very interesting actually data set. I have a question which is more or less what you discussed as well on the perspectives. Through the years you experience all these transitions that we have on the algorithmic point of view. So you have also to, I saw that you adjust the data sets, you added more data and you upgrade it, but did you have also to adjust the evaluation kind of protocols that you had in order to evaluate the performance of the algorithms, or?
Let me show you a very simple, we had to change over the years, yes. Let me show you a very interesting example. If you look at the yellow line here, there's a big drop in the middle.
Yeah. 2000 actually.
Yeah, so everything seemed to be perfect up to a certain point, and the reason for this perfection was that the balance between yes and no questions was not equal. So there was a bias that we didn't foresee at the beginning. And when we changed that, things changed. This is a very simple example. We had to change more. At the beginning, for instance, we included things that had to do with annotating with concepts, ontology concepts. This ended up to be very difficult for our experts to handle. But regarding the methods themselves, I don't think we did any changes to the evaluation protocol because of the methods. We developed a new method for hierarchical evaluation of the multi-label text classification, but that's it. We didn't do anything regarding the method, based on the methods.
Okay. Maybe I can follow up on this question from your experience over all these years. Have the questions become more difficult? Do you think we will ever get to a point where biomedical questions can be answered by systems like this with high fidelity?
Regarding those questions, I'm pretty sure we can get better.
Okay.
And we are already in a decent stage. Actually, one graph that I didn't show is how, during the manual assessment, what do the experts think of how good the questions are?
The questions.
And actually, the opinion of the experts about the questions is very high. So irrespective of whether they get the right documents, they get very good questions, very good answers. So maybe I should be closer to the microphone. So they get very good answers. The experts are very happy with the answers. Regarding the open questions, things are a bit different because these are questions that the experts don't know the answer to, right? They're more difficult. I do believe we can get very good at that. And I also think that we can get better to what they currently have, from what they currently have. What they currently have is very poor. They miss a lot of very useful information, or they have to spend hours to find it.
They is
The expert, the clinical doctors or biologists. Yeah. So I think we can help them a lot, even by having imperfect results.
Yeah, and many people want this as a consultation tool, not as the ultimate
Yeah.
decision tool.
We're not talking about automated decision making.
Okay, one more question.
You did this is wonderful, very interesting example. You ingested literature, papers, and so on. But you consider also ingesting and cross-referencing clinical reference libraries like the UpToDate library, things that clinicians use day to day to pull out information within the practice.
Well, the UpToDate library is actually what I think is the current state of the art. This is what I was comparing to before, yeah. We haven't tried that. It's very manual, it's a lot of work, you cannot scale, yeah. But we haven't tried to combine with that, it's interesting.
Very interesting cross-reference.
Yeah, true.
Okay, please join me in thanking Ms.
Thank you. After this first talk, we are going to move to Maria Papadopouli. Maria Papadopouli is a professor in the Department of Computer Science at the University of Crete, a research associate at the Institute of Computer Science IV, and a Fulbright scholar. Before going to Crete, Maria was an assistant professor at the University of North Carolina at Chapel Hill. And since she has been in Crete, she has been a visiting professor at the Brigham and Women's Hospital, the Harvard Medical School, the Computer Science and Artificial Intelligence laboratory at MIT and at the School of Electrical Engineering at KTH. She is a very prolific researcher. I first learned about her work in networking, but being the curious person that she is, she has moved to different fields, and her talk today is a proof of that. She has several awards, the IBM Faculty Award, the Google Faculty Award, Comcast Innovation Fund, and funding from competitive national, EU, and international grants. Please join me in welcoming Maria.
Thank you very much for the kind invitation. I'm delighted to be here talking with you and learning from you. So today my talk is going to be a little bit different. I'm going to present a joint work with Stelios Menakis and Yannis Birnakis on identifying neuronal modules of communication. So we are all impressed by what we hear with the developments in AI, autonomous driving, language models, and sometimes actually in the news we get also some events like having this kid taking his grandfather's car driving to the candy store. So we said, wow, I mean, how this is possible. And developmental psychologists provide these charts that show these amazing things that a baby can perform. How can babies learn how the world operates? How they can get this sense of stability, of robustness, do this face tracking? So I was actually listening to an amazing talk by Joshua Tenenbaum at Brain and Cognitive Science at MIT, a very inspiring talk where he was describing his vision about trying actually to model the world and agents and have this intuitive physics and intuitive psychology in this modeling framework, and essentially performing a kind of reverse engineering of the common sense core. So this is a timeless question. How does the human brain obtains so much from so little? And obviously, a timely opportunity to try to develop more efficient, more effective, energy efficient and robust AI systems, bridging the neuroscience with AI and engineering. And obviously, computer science can play a very important role here. So the rapid advances in optical imaging, in machine learning, and all this availability of computation and storage resources provide the unique opportunity to decipher this fundamental question, which is how does the brain perform the complicated computations that allow us to learn and interact with the environment? And the aim of my lab and the team of collaborators that we have is to try to improve our understanding of the neuronal circuit function, and then try to incorporate all these findings in ANN and neuromorphic computing hardware, aiming actually to try to develop a new generation of computing technologies based on the organizing principles of the biological nervous system, which is optimized for higher level of cognition. So today, in this talk, I'm going to try to present these efforts. And the video here, which I believe it was shown, right? It is from 2-photon mesoscopic imaging tool from the visual cortex of a mouse. This is a video that was provided by Stelios Menakis, our collaborator, and you could see actually the neurons in the visual cortex firing. And well, machine learning and ANNs and neuromorphic computing actually have been greatly inspired by all the developments and all the discoveries in neuroscience since 1906, where there was this Nobel Prize to Cahal. And basically, these are two parallel paths that have been influencing the sciences. So we know that brains are machines probably the most remarkable machines ever built, and they perform computations, they have circuits, actually they run algorithms, and they actually perform multiple processes unfolding in different space-time organization, quite efficient, and they have been affected very much by adaptation via evolution, development, learning, thinking, culture. And they have mechanisms that adaptively build new architectures and new mechanisms. So this is actually a new era in synergetic neuroscience and AI, for which I'm really enthusiastic. So we know that in the brain there are up to 100 billion of neurons, and each one has up to 10,000 synapses. So this is like 10 to 13 of computing elements, propagating signals, and neurons actually forming circuits, sharing information, propagating activity, generating and propagating activity from one layer to another, from one area to the other area. And you can do the analysis at different scales, microscopic, at the cellular level, and this is what we are doing, analyzing the firing activity of neurons, or at mesoscopic or at macroscopic level, where we have multiple columns, we have the interaction across different areas. So what functional analysis tries to do is identify the organization modules which essentially will provide clues about the operations that they are performed in the brain, in the cortical networks. So the work today, the results that I'm going to present today, are based on analyzing the functional connectivity in the absence of stimuli. So it is under spontaneous conditions. So the mouse was quite awake on the treadmill in a dark room without any stimuli. And the functional connectivity inferred from the ongoing spontaneous conditions reflects functional relations between two cells that could be anatomically connected or so directly connected, or shared input that these two cells have from another cell. And there is a very inspiring work analyzing the functional connectivity from the community. So today, in this talk, I'm going to present two threads of research. First, I'm going to characterize the functional connectivity using graph theory. And then I'm going to actually present the main building block, the main communication building block from layer 4 to layer 2-3. So let me actually first give you a sense of our data. So this is a figure that you can see 3 neurons from visual cortex of the mice. And the data that we are analyzing are essentially, as I said, eventograms from thousands of neurons. So these eventograms indicate when, at which frame, and the frame is approximately 100 milliseconds, the neuron fires. So these data were actually provided from Tolia's lab at Baylor, now at Stanford, where essentially he recorded with this 2-photon mesoscopic imaging tool simultaneously from thousands of neurons from 5 mice. And what he records is essentially video, which essentially is the fluorescence, the pixel intensity. And this signal is deconvolved, so we get the probability of firing using Pneumatikakis algorithm, and then we actually threshold this signal to get the eventograms that we are analyzing. So here you see basically the hierarchy in the V1, in the primary visual cortex. So each dot is really a neuron. So you see here 3 layers at different depths. Layer 4 is considered like the input layer of the visual cortex. Layer 2 and layer 3 are quite similar, and that's why you'll hear me talking about like the output layer, layer 2, 3. And I'm going to first try to present the functional connectivity in the intralayer. So the functional connectivity is really based on pairwise correlation of the firing events of neurons. So let me actually describe this correlation. So we use the spike time tiling coefficient, or STTC, a metric which has many advantages over other metrics like Pearson or entropy, etc. And what it does really is assess the synchronicity of the calcium eventograms of the spikes of neurons. So it is based really on computing the PA or PB, which is the proportion of spikes of B within a lag of the spikes of A, and then subtracting, discounting, essentially the correlation detected by chance. So basically reducing with the proportion of total recording time from the spikes of A, and then appropriately normalized and symmetrized. So we use STTC. So basically, let me actually show you here. So you see here in the green, layer 4, the input layer, the reference neuron layer 4, and its neighbors at layer 4 also. Intralayer connectivity, functional connectivity. So let me actually describe these results. So there are statistically significant functional connections. So it's approximately like 23% of the connections are statistically significant with a conservative Z-score of 4. And at the left, you see that the STTC values, the correlation that we found in the visual cortex, is statistically significant. You see the difference from the null, which is when we are circularly shift the spike trains. And in the middle, you see actually the STTC of the statistically significant edges. But however, keep in mind that the temporal correlation that was found is low, and actually STTC is a pretty robust metric. We found similar values under different time intervals, under different spatial lengths of the connections, and also other different conditions. And then we studied actually how the statistical significant edges change with the edge length. So as expected, there is a bias to locality. So there is a bias towards short functional connections. However, there are even long, strong edges. Another interesting thing is what I just mentioned to you earlier, that you can observe that the STTC is pretty robust for different edge lengths. So if now we apply very simple graph theoretical metrics, we can actually start forming the simplest unit beyond the cell, beyond neuron, and which is the neuron with its neighbors. And you see that the normalized degree of connectivity up to the 0.6 value has like a uniform distribution. And then we also estimated the cluster coefficient. So the cluster coefficient actually being careful to remove the frames where the reference neuron fires so that we don't have this bias. And we found that actually the cluster coefficient is high, it's pretty high, it is like 0.6. So neighbors of a neuron being connecting with each other reinforce the idea of having like a cooperative unit. So what we see here is that layer 4 does have a structure as it is reflected by the degree of connectivity and the cluster coefficient. And then we compare the function connectivity in layer 2-3 with the one that we found in layer 4. So compared to layer 4, layer 2-3 has smaller number of statistically significant edges, has smaller cluster coefficient, slightly less STTC weight, and smaller number of statistically significant edges. And we wonder, is this differences due to sparsity? It is interesting also to observe that layer 2-3 has a different distribution in the degree of connectivity than layer 4. And we're wondering why nature wants this. And you could think, these are now speculations, but you can try to assess them, if this is due to the ability of learning better. In layer 4, you want more like a uniform distribution so that you can apply different size invariances. And in layer 2-3, you may want to have more abstractions, easier generalization. So these are important questions here. Similar results also we found when we are studying the interlayer connectivity from layer 4 to layer 2-3. But now let me move to another important concept, which is how actually the activity is propagated from layer 4, from the input layer, to layer 2-3. So basically you can see here the main communication unit. So basically you have a recipient, a putative recipient neuron in the output layer with its connections from layer 4, all its neighbors from the layer 4. So this is an important, in our opinion, communication unit that we'll be studying in the second part of my talk. So what we found is that actually we can predict the firing of a neuron at layer 2-3 based on the number of co-firing events at layer 4. Not only that, that this is really can be modeled well with a ReLU. And you can see here the R² fit of the ReLU for our neurons. Now, it is interesting also to observe the difference of the response with the null, where the null was formed by a group of equal size as the observed STTC group, but randomly selecting the neurons from the visual cortex with the condition that they don't belong in the layer 4 STTC group of our response neuron. So how we read this plot, which is a very simple but very important in our research result. For example, you can see here that if at a certain frame, there are, let's say, 34 co-firing events from the specific STTC layer 4 group of that reference neuron, then the probability for this neuron to fire is 0.8. And you see the standard error of the mean. So then the second important, in our opinion, rule that we discovered is the fact that actually the response of the neuron gets reliable rarely. So what we did here at the plot at the right is, it is a scatter plot. So we took the probability of the neuron at the layer 2-3 layer to fire, and in the y-axis, and at the x-axis, you see the CDF of the co-firing events. So it's a scatter plot based on the plot at the left and the CDF of the co-firing events. So what you can observe is the presence of two regions. So actually the first region is the region of non-response, whereas the second region is where actually the layer 2-3 neuron start responding with a linear trend. It rises linearly. So this is interesting, and it is kind of like discovery, the fact of that, number one, it is a rare event when the neuron responds, and that there are these two regions, distinct two regions. And this is across actually all, a lot of neurons. Actually, in this specific histogram, you see approximately 15% of neurons that have the following property are our good responders, meaning they have a maximum firing probability that is above 0.7 and a reasonably good R². So you see that the intersection of these two lines is approximately 0.9. All right, so then we're wondering, okay, we found that there are neurons with different degree of connectivity, and there is this entire continuum. So it's not like there is a dichotomy of only very small and very large. There's the entire continuum, but how these neurons with different degree of connectivity respond? And what we observed is that actually small groups are more efficient in transmitting the activity from large groups. And we're wondering why. So when we saw this plot, and I can explain the yellow line here in the response corresponds to neurons with small degree of connectivity at layer 4, so few neighbors, and the purple line corresponds to neurons with large size, a lot of neighbors. When we saw this plot, we said naively, well, if you normalize things, so instead of having the number of co-firing events, you take the percentage of co-firing events, you have the lines being coinciding. Well, things in nature are a little bit more complex. So what we found, and this is like the third finding here, is that we need to divide the number of co-firing events to the group size, but at an exponent. And this exponent is consistent across all mice that we had, and it is around 0.6. So we're wondering why this is the case. So here we have a very simple theoretical model to describe that, which is actually just based on binomial distribution using the central limit theorem, where you see that if f is a number of co-firing events, then you can do this just simple mathematics, and you could find out that the response probability is really proportional to the number of co-firing events divided with the square root of n. And why square root of n and not 0.6? Well, things here are not completely, they are not exactly Gaussian, right? So now we start thinking, okay, we know that in the brain there are different internal states, there are different brain states, right? And we do have two proxies for the internal state, namely the population activity and the pupil size. And you know, guys, the pupil size is a proxy of the alertness. The larger the pupil size, the more awake, the more alert is the animal. So we said, let's try to observe our neurons with respect to the different internal states. So what we found, and this is not really surprising, that was like the first related result, is that layer 2-3 neurons of large groups are more modulated to the population activity. What you see here in this plot is that you see these two clusters, right? The first, the two extreme, with the smallest degree of connectivity, the yellow, and the largest degree of connectivity, the purple, with their ETA signature. What is the ETA? Simply, it counts for each neuron, what is the average population activity with respect to its firing event, to its calcium event, right? So it's like a signature, a very simple signature. So what you observe here is that unlike neurons of large groups, the purple, neurons of small ones, the yellow, are less modulated by population firing. So in this concert, in this symphony of our brain, the neurons with large groups are like the choroists, whereas the neurons of small groups are like the soloists, right? More independent. And we did find this independence in different also threads of this research. And this is a kind of like consistent result with what other researchers know. However, more interestingly, what we found is the following, that if now we take a neuron that has a small neighborhood in layer 4, and we take this neuron of layer 2-3, and we take its eventogram, and we compute the Pearson correlation with the pupil, what we found, and you can see at the left plot, is that it has positive correlation values, right? There are a lot of values around 0, but there are also positive correlated values. Now, when we go to the other extreme and we focus on large groups, layer 2-3 neurons with large neighborhoods, and we take the eventograms and we take the Pearson correlation, the pupil size, what do we observe? Negative values, which means that when neurons with large groups fire, they fire when the pupil size is small. There is negative correlation, which means that the mouse is not alert, is not awake. So let me now summarize our main results here. There is substantial functional connectivity, pairwise connectivity, both in layer 4, layer 2-3, and in between. A reasonably long-range functional connection, strong, up to 1 millimeter. The hub structure in layer 4 is different from layer 2-3, and there are very interesting theoretical questions. We can predict the firing of layer 2-3 neuron based on the co-firing events of their group at layer 4. The response can be modeled as a ReLU quite well. We discover these two regions in the response function of the neuron, and that the neuron at layer 2-3 responds with a linear shape in rare events, after a certain number of co-firing events. But these are rare events, which is important to know. Moreover, small groups, layer 2-3 neurons, smaller groups are more effective in transmitting the activity downstream. than large ones, and layer 2-3 neurons with small layer 4 groups appear to behave differently than those of large ones. And we believe that this functional analysis identifies organizational modules which contain clues about the operations executed by the cortical networks. And this is an attempt to move from the cell unit to multineuronal principles of activity generation and propagation. So I just would like to say that we are currently performing similar work under different conditions, under conditions of stimuli presentation, learning, and when there are neurological diseases, because we would like to build neuronal biomarkers to observe the progress of a disease. And of course, this work on spontaneous conditions can serve complementary to the research of direct anatomical and physiological cortical connectivity. So thank you very much for your attention, and let me acknowledge also the great help of talented undergrads mostly actually, and some graduate students in my team at Forth and at the University of Crete, and also the valuable collaboration of several people, many of them from Greek diaspora. So thank you very much for your attention. Thank you.
Okay, questions? Yes.
Thank you very much, Maria, for this wonderful presentation. So you alluded at the beginning of your talk that there is a certain way that baby brains work and a certain way these large language models work. So after seeing your presentation, are there thoughts about creating algorithms or neural nets with activation functions that are inspired by these findings, and for what kind of problems may these be relevant?
Yeah, that's a great question. Thank you for that. So actually, this is what in my team we are trying to do currently. So more specifically, we are trying to find ANN architectures, for example, that they are bio-inspired and work well, and they are pre-trained, and then try to take this find. First of all, we are trying to see whether we can observe in these architectures, this finding, these principles, these rules that I just presented. That's the first step. And if we don't see them, we are trying to modify the architectures to essentially adopt, to integrate these rules and see how the performance changes. So this is the first step. The second then step is when we are happy with the results, that we feel that, you know, in terms of the prediction that this architecture tries to do, because it serves a specific role in the, let's say, in the vision domain. And we can see that, you know, integrating our findings, this does not sacrifice the performance. After do this, the next step is to try to see how we can run various hypotheses that can then help us to design experiments for the neuroscientists. So basically, this research tries to create a loop, which is we try to apply machine learning and AI algorithms to analyze our data. This is the one more direct thread, but there is also a backward link, which is having an ANN architecture, which is bio-inspired, that integrates all these findings, which we can run and to test hypotheses before going to run the experiments that they are quite expensive.
Thank you.
So I'm not sure if I answer your question, but yeah. Okay, thank you.
Okay, do we have another question from the audience? All right, thank you very much,
Yeah,
Maria.
thank you.
Okay, this takes us to the last talk of this session, and our last speaker is Maria Vakalopoulou. Maria is an assistant professor and the team lead of the biomathematics group at MICS laboratory of the Central Superlek University, Paris-Saclay, and she's also affiliated with Paris-Saclay. Prior to that, she was a postdoctoral researcher on the same university. She received her PhD from the School of Rural Surveying and Geoinformatics Engineering at NTUA, and also she received her engineering diploma there. Her research is focusing on artificial intelligence, on the development of methods and algorithms for visual perception, including remote sensing and medical imaging. She has led, one of the characteristics of her work is that she works in close collaboration with clinicians in France, and in particular in the Gustave Roussy and the APHP hospitals. And I will let her talk about her work. Let me mention that she has published extensively in several conferences and journals, and also received many awards and grants. Maria.
Hello all, thank you very much for the invitation, and I am very glad to participate in this very, very interesting workshop. So today I will talk about some of our research, but also things that I think they are happening now in the medical field. So I will focus on artificial intelligence and how we can use these tools and algorithms on medical imaging and precision medicine. So I will start with a definition of precision medicine. So actually what we are trying to do is identify, actually not us, but clinicians, they try to identify phenotypes of patients in order to see how they can perform well on different kinds of treatments and have some kind of prognosis. And currently actually there is a lot of work that shows that all this kind of field can benefit a lot from AI algorithms and AI advances, that they can help actually understand also better what the human health and how we can actually characterize and create this kind of groups and clusters. And indeed these works, they are currently gathered the attention of very high impact journals, like for example Nature Science, that they show that indeed all these algorithms and tools, they can really benefit having a better and helps with clinicians with their everyday practice. And however, when we are talking about these tools, and of course we're working on the field of medical imaging and medicine, we have actually to tackle quite some different challenges that they are very important, and they somehow constrain the way that we can actually develop these algorithms. So let me just mention some of them. First of all, we have to actually work with limited data, that they are either small in size and quite imbalanced in terms of what they represent, since most of the times the anomalies, they are quite smaller with respect to the general population. And also for this data, most of the times we don't have access to all this kind of very expensive and domain-specific labels that we need from the clinicians. So actually we need somehow to tackle and create algorithms that they are trained on limited amount of data with limited amount of annotations. And moreover, even if we have these annotations, we know that these annotations, they can vary a lot. Since different doctors can have different opinions about particular cases, or even different hospitals, they can have different protocols that they acquire their data for the particular clinical practice. So all these add a lot of limitations and additional problems on how we can build algorithms that they are really data specific, and they are actually able to be trained and harvest all this knowledge that we have from the data. And of course, all this comes also with biases in the data sets that somehow we need to be aware or find out and try to cure in order to create algorithms that they can actually work very well on different kinds of cases. And finally, something that is also very important is that all these algorithms, most of the times it's very difficult to have explanatory power, and that means that they can work, they can give us very nice performances, we can evaluate it with different metrics depending on the task, but at the end, when we are talking about problems that they have to do with very important problems, like for example deciding for a particular kind of treatment for a patient, we need actually to know how these algorithms, they base their decision on, and be able to interpret these results in an analogous way. However, through these different years, medical imaging follows the advances that they are happening actually in the computer vision, in natural language processing, and all this kind of communities. And actually, there are also a lot of data sets that they are published more and more, and they are open access, and people can actually use them either to benchmark their algorithms or to train their algorithms. For example, we have the huge TCGA portal that contains a lot of data related to cancer patients, or we have actually more imaging data sets that they are coming from grant challenges that every year the community organize in order for the engineers to benchmark their performances. And of course, we are when we are talking about these variations on the annotations of the protocols, we are talking also about models that they can actually, we are trying to build models that they work on different modalities. So we are trying to see how we can do a good transfer learning, few shot, zero shot learning without actually having access on a huge amount of data. And the way that the data set can be built, this is actually an open question. There is a lot of discussion currently on the community how we can build all this data that they are unbiased and feel free of all these kind of variations, and they can build algorithms that they have very good generalizability on variety of other modalities. So we can use annotations from different experts. We can have actually more merged data sets, work on the problems of domain adaptation and so on. And finally, in order to have this interpretability, it is of course a very huge field, but of course we can, people they are trying to adapt some kind of attention module, so try to identify somehow how this important decision comes from these models. And actually, in our group, we are focusing on cancer. So our work is mainly focusing on different kinds of cancers. And why cancer? Because it is one of the leading causes of death worldwide. And also, one more thing is that this is a disease that we don't know a lot, or we need to explore more, not only on the methodological side, but also from the biology side. We know it is a very complex disease, and there is a lot of things that we need actually to understand more from the medical point of view as well, in order to help patients and create somehow treatments that they have better outcomes. And since this kind of disease is very complex, what we have noticed the last years is that we don't need actually to focus on only one modality. For example, images that they are coming from radiology, that they contain information, but of course, we cannot contain all the information we need in order to build our knowledge. So we need actually to create algorithms that they will be able to use a very big different amount of information that comes from different scales, from radiological images, from genetic information, or even from digital pathology images, which are actually huge images that they are taken from tissue biopsies. And when it comes to that, we need to keep in mind that actually this data, they are very high dimensional and they are very heterogeneous between each other. So how we can actually build data-driven algorithms, having also access to limited amount of annotations and very high quality data set, it is actually a very open challenge. But for my talk today, I will try to focus mainly on the work that we are doing on the digital pathology part. And why digital pathology? Because actually it is the golden standard that we use currently in order to do a kind of diagnosis of cancer patients. This is actually the way that doctors decide about the type of the cancer that a patient has. And it is done in a way like the patient needs to go under a surgery, and then the tissue that it is removed, it is analyzed under a microscope, and then the doctor needs to actually create a report summarizing what he finds. And actually, only very recently we can have access to this kind of data in a digital form. So there are a lot of actually from 2017 and later, we can access this data, and we can actually think about creating tools that they can either help the entire process and automatize parts of it in order to make everything much faster. And how we can do that? The AI community the last years has a lot of advances. We are talking about a lot of models that exist, especially when we are talking about images. We know that convolutional neural networks, they are there for more than 10 years now, so they can give us both very nice representations that they are learned directly from the data, as well as some performances that they are very impressive with respect to what we can have for these different problems. And of course, since medical imaging follows also what is happening in the vision community and the natural language processing community, we have also transformers or visual transformers that they start also being integrated as very powerful models that we can use for the representation of this data and the final clinical endpoints that we are looking for. And these models actually, more or less now, we have a kind of standardization of the process that we use in order to analyze digital pathology slides. So more or less what we do is that we take these huge images that they are of gigapixel size. So you can imagine even storing a data set, it is challenging. And what we do is that we create some, since with the current hardware that we have, we cannot really use it as a whole. We create some kind of patches that they are extracted from all these different kind of the different regions of this of the digital pathology slide, and then these patches they are passed through this kind of architecture. You can think about any architecture you want to use depending on the recent advances, and then they are used in the multi-instance learning setting where actually we can combine somehow all this representation in a meaningful way in order to predict the final endpoint that we want to have. For example, classifying different subtypes of cancer given the particular kind of data. And actually in 2021 there was a paper on Nature Biological Engineering that gave all this kind of pipeline and automatized the entire process, and this is actually what it is used currently as a baseline to evaluate algorithms. However, something that we need to acknowledge here is that even if this pipeline works very well, most of the times the representations that we are using, they are coming from pre-trained networks that they have been trained in a completely different domain, most of the time ImageNet or any other kind of huge image-oriented dataset, and they are used as a plug-and-play model in order to create the representation of these tiles, and they work very well. However, they are task and data agnostic. That means that they haven't actually trained neither for the data that we are working on nor for the particular task and endpoint that we are looking for. So actually the community has worked a lot on how we can adapt these models and how we can train them at least on the data that we are working on. And here there are a lot of methods that they are focusing on self-supervised or unsupervised learning, where we can actually try to find tasks that they don't need the data set, but we have access actually to all these big pools of unlabeled data and use them as a pool of data that we can train or adapt our algorithms for the particular application. And actually in our group we are focusing a lot on that, and we are trying to develop unsupervised or self-supervised methods that they work well both on classification tasks as well as dense tasks, like for example semantic segmentation, as to how we can segment cells from different kinds of input tiles. But of course now the current trends, and what I guess we have discussed also a lot in the previous session, is that the community in general goes more and more to this kind of large models and how we can use them and how we can actually adapt them. And indeed, the last 2 years, I would say that these models, they perform very well. They give results that they are impressive. We can do things that we couldn't do before with such a good performance. As for example, here you can see two algorithms that they have been used in order to generate images from text. And indeed, you can see that the images that they are generated, they are really, really realistic. And this is quite impressive. However, again, these models, they have a huge amount of parameters, 12 billion parameters, and they have been trained to these huge data sets, the LION data set that Alex was mentioning before, that it has millions of pairs of data, of course, that we can definitely not be able to access in the medical domain. It's impossible to find annotated data sets of this scale. So, of course, there were a lot of people that they were wondering how we can use these models, since they are so powerful, in order to somehow solve medical tasks. So actually last year there was a paper in Digital Medicine that tried actually to use this glide model, this generative model, out of the box, just from the API that the OpenAI was given, and try to generate images given particular kind of text prompts, as for example, please generate to me an MRI of the brain, or for example, an X-ray of the lungs. And what actually they noticed is that even if this algorithm had impressive results in all these kind of natural images, here the performance, it is much, much lower, and what we get, it's actually not actually we want in order to say that this kind of models, they work well. And in order to evaluate somehow, because here you can see some of these examples, but actually the authors, they try to do an quantitative evaluation. So they ask some experts to annotate how realistic are the images that they are produced, and what actually they saw at the end is that for modalities that they are, for example, focusing on histopathology or scientific papers and scientific illustrations, the generations, they look quite realistic, and I could say that this also maybe covered more on data that they are publicly available, and maybe they are already included on the training of these algorithms. However, when we are talking for more medical modalities, coming, for example, for radiology, as for example, X-rays, MRIs, CTs, all these generations, they were not really realistic. So actually, it shows that we need actually to build tools that they are able to adapt on this kind of, on the medical data that we have access and we want to use. And why actually this is important? Because actually, we can even think about using these algorithms in order to ask questions, like, for example, generate to me a histology image of a patient with benefit from immunotherapy, and then try to see, with respect to this generation, if the characteristics that we can have, they are matching with the characteristics that we're using currently in order to identify patients that they go under immunotherapy treatment, or even adapt it or change it with respect to try to analyze why this is happening. So indeed, even if this can sound now a little bit unrealistic, I really think that these models, they can give a lot of insights on biomarkers that we know, or even we want additionally to explore for the different kind of problems that we are working on. And of course, there is again the ChatGPT, a huge model trained again on a huge amount of data. Models that we cannot even in the university be able to train, not even we cannot even store all this kind amount of data that we need for this kind of algorithms. And of course, again, there is literature on how we can use these tools, as for example, the BioGTP that it has been mentioned before, in order to answer or do some kind of diagnosis giving some kind of symptoms and see what actually can be the best things and treatment to follow. Of course, all this is an open question, and we need to actually interpret it with a lot of cautiousness. But there are all these tools; they are there, and it is actually an open question how we can use it on the medical domain. So actually, in our group, we did recently a work on which we try to use this ChatGPT in order to do some kind of data augmentation of text that has been generated from us, integrating some kind of clinical attributes that we have for each patient. So what we did is that we asked the doctors to create some templates, some sentences that they contain the clinical attributes that we want to encode, and we use these sentences as an input to ChatGPT, asking it to create different text variations. And why this is interesting? Because like that we can enhance our data with more text information that we know that somehow it's more informative than one particular attribute that we can have for a particular clinical attribute we want to integrate. And indeed, what we notice is that we train this together with the imaging modality that we have for predicting failure of kidney transplant. And what we saw is that indeed we could perform better than other multimodal algorithms that they are able to perform the same task. So indeed, there are a lot of things that can be explored on this kind of direction. And one also recent model that it has been proposed by Meta very recently is the SAM, or Segment Anything algorithm. That it has been again a huge model that has been trained on 1 billion masks from 11 million images. And actually what it does is that it can give very accurate segmentation of objects into images. And indeed, we know, and it was very impressive, and there are already things that show that this kind of algorithm, they can be adapted on data, as for example digital pathology, on which maybe it cannot actually perform very well as the SOTA that we have currently, but it can reach very impressive results with respect to how it has been trained. And how all this work is, and how actually the community started working on is what we call now prompt-based learning, which is actually a new way of training or adapting the models or these large models that we have in data that we don't have such a huge amount of data and labels. And what actually was happening, and it has happened all these years, is that we do this kind of traditional fine-tuning strategy on where we have usually a model that has been pre-trained in another data completely out of the domain, and we use some kind of additional parameters in order to adapt this data to the task that we want to address and also to the data that we have to address. But when we do this pipeline, most of the times we need again to parameterize a lot of parameters that we don't have data for. So with the prompt-based learning, what we do is that instead of having this tunable part, we can insert a prompt together with the input into the pre-trained model. And this prompt can be anything that you can think of: a text, an additional image, variables, parameters that you actually learned through the entire process and train it for the particular task that we want to solve. And indeed, it has a lot of advantages. And then the traditional fine-tune setting. And this is actually what we did as well. We tried to use it and adapt it on processing of digital pathology slides. And in this setting of multi-instance learning, where we have all these patches that we want we don't have the label for, but we want to use in order to perform the final label. And what we did is that we insert these parameters together with the inputs. And these actually, they have been trained together with the classifier in order to perform the task. And indeed, by just adding only a very, very few amount of parameters, we were able actually to perform very well. And we did actually experiments on 3 different public available digital pathology data sets, all of them that they have to do with classification tasks, either in breast or colorectal cancer. And what actually we saw is that we have a very consistent gain of performance with respect to traditional fine-tuning strategy or even to conventional ways that we use pre-trained models with. And of course, this kind of setting, it's very easy to be adapted on any kind of multi-instance learning algorithm. So it is very, very interesting to see how we can boost performances even more. But also, one thing that I am very excited on is not only about the performances, because we said that we need actually to interpret the results, so we need to know where actually the algorithm is focusing on. So what we understood is that by having these parameters together with the inputs, we will adapt the way that, or guide better the regions that the algorithm is based on, and actually the attention maps or the attentions that the algorithm is paying more attention, it seems that they are more informative with respect to what we are looking for. We can see here that we are able to attend more on cancerous regions, which is what actually we want at the end to have as a label. So, in order to conclude this presentation, of course, currently we are in an era that AI achieves very impressive results, and actually this actually is translated as well on the medical imaging and clinical oncology. All these kind of large models, they are there, and I really think that they will play a significant role for the research in the future. And what I think it was, what Alex also mentioned in his talk, I really think that how we can adapt these models on the tasks that we have in a good way, and with all these limitations that we have, depending on the domain, is a very, very important field of research, and this is what I think people will focus on. And yeah, recently I saw this cell kind of front page and saying that the cancer is actually very complex, and it is a quite interdisciplinary area that we need all these different kinds of expertise in order to be able to progress rapidly with the research. So we are, in our team, we are trying actually to pick one of these strings and solve this problem. So that's all from my side. Thank you very much for the attention.
One or two questions. Please.
I think this was excellent, covered so much ground, so very well. Just a question about the
Okay.
we tried it with CT, MRI, and ultrasound, and nice scores, F1 scores, practically down to 0. You got good results. Can you comment on that?
Yes, very good question. Thank you very much. Indeed, some was out, and then everybody started tweeting about that, and of course, people starting also using it, especially on CT scans, MRIs, and everybody was saying, "Oh, it doesn't work. It is not working very well." And of course, one thing that we should keep in mind is that the way that SAM is working now doesn't have this semantic segmentation meaning. It just segments you objects. These objects maybe they are not even meaningful for your task that you want. But also, if the image is very out of distribution, it fails also completely. And this is why the prompts that you can insert, if you use it like that without inserting anything, definitely I think it will fail. But if you try to somehow either add prompts so that they are meaningful for your task in a manual or even automatic way. We are working on how we can actually create this automatic way to generate the prompts, which can be dots, boundary boxes of objects of interest. It can actually work quite well. And we have tested, for example, on segmenting organoids on microplane, yes, and it works very, very accurately. Also, one thing is that working it out of the box, again, it will fail. You will need to find ways to adapt it. This is what we try to do in this project, on which we somehow add some additional prompts that they are able to adapt what this kind of huge model into the data and the task that you want to solve, or even perform semantic segmentation with respect to that, because you can even integrate classes. I agree. Having it out of the box, especially for data that we are working on, it will most probably do not work, but there are ways that you can somehow help you make some use of it. I don't know if it will be better than the current tools that you are using, either in clinical practice or the state-of-the-art algorithm that you are using for the segmentation of the organ that you are looking for, but definitely there are ways that you can adapt it, and it will be at least somehow interesting to see if it works or not well for our community. So.
Okay, do we have another question? Okay, let me follow up with the question. Do you see sensitivity on where the data comes from, or you've not gone into this level of analysis?
No, this is actually, yes, this is one of the problems that we have, especially in digital pathology. Even if you are using, the hospital are using the same kind of staining for the tissue, this staining can have a different variation of the intensity, and having data from a different hospital can actually fail the performance. So there are even challenges that they are trying to see how we can adapt, we can use domain adaptation methods in order to somehow use either unlabeled data from other sources, or even try to create algorithms that they have a better generalization with respect to this data. So definitely there is a very big sensitivity on the protocols that each of the clinical center is using.
So a lot of work to be done.
Yes, a lot of work to be done.
Okay, any other questions? All right, let's thank Maria.
Thank you.
Okay, this concludes our session, and we are going to meet again here at 4 o'clock. Thank you.